Hello, everyone, and welcome to today's webinar, Trace Level Cytokines and Cardiovascular Profiling for Long-Term COVID Diagnostic. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Nanion Technologies. To learn more, visit www.nanion.de. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. We do also have a group chat feature located on the right side of the screen. Feel free to interact with our speakers and each other during this webinar. I'd like to now welcome our speakers, Sonja Stoelzle-Feix, Director of Scientific Affairs and Product Manager of Cell Analytics Systems with Nanion Technologies, and Kirsty Short, Associate Professor, School of Chemistry and Molecular Biosciences with the University of Queensland. Sonja, you may now begin your portion of the presentation.
Hello all together. I'm happy to welcome you to this webinar today. And before we get started with Kirsty's presentation, I'm happy to walk you through some brief background information on some of the topics that are being addressed today. So, for example, the immune response triggered by SARS-CoV-2 infection has been implicated in the development of long COVID. And cytokines, the cytokine network ensures a coordinated and effective immune response against these pathogens. Furthermore, immune dysregulation may contribute to cardiac complications observed in some long COVID patients. So, how can we investigate these cardiac complications? So we can actually make use of tools and models that are being already developed by the cardiac safety communities, for example, the Safety Pharmacology Society. Safety pharmacology is a distinct scientific discipline that integrates the best practices for pharmacology, physiology, and toxicology. And as most of you also know, is that safety is a critical component of medical product development and approval. So due to the work done here, withdrawals of new drugs due to side effects is diminished. So one cell model that is heavily used over the last years are induced pluripotent stem cell-derived cardiomyocytes, so iPS cardiomyocytes. More than 10 years ago, Yamanaka and al. Earned the Nobel Prize for the discovery that mature cells can be reprogrammed to become pluripotent. And this changed a lot, so also for cardiac applications. Now, looking at peer-reviewed papers during the last years, we found that about 100 to 150 groups publish at least one paper per year. And this is distributed over 70 countries and over 20,000 researchers. This is basically the top 10 here. So the US leading, then comes China, Germany, and Japan, and so on. So indeed, and I'm speaking about the cardiac safety community here, we've come a long way. So recent publications looking at electrophysiologic properties of iPS cardiomyocytes and native cardiomyocytes, and publications around best practices, benchmarking new protocols have been published. So basically all of these authors here aim to provide guidance and answers to critical questions that matter to patients, regulators, and scientists. Now, let's have a look at some methods where these iPS cardiomyocytes could be investigated. So, just some examples here. There are, for example, the optical methods, imaging. These are kinetic measures, so you get multiple data points over time. It is a real-time method, but here you have to use dyes and reporters, and it is most likely destructive to the cells. You also have the so-called endpoint assays, where you only get a single data point at the end. This is very time-consuming, and it is also destructive to the cells. Now, there are also plate-based assays, for example, impedance-based assays. And here you get also kinetic measures, so you get multiple data points over time. It is real-time, label-free, and non-destructive to the cells. And actually, this is where Nanion, this is the company I work for, comes into play now. We were following down this road since more than 20 years now. We are developing automated patch clamp plate-based systems and also impedance-based systems. So I would like to show you one example here. This is the Flexite system, which has a silicon substrate based well plate as a recording consumable. You see here in the video silicon membranes in a 96 well plate, and on these silicon membranes you have iPS cardiomyocytes sitting. These cardiomyocytes lift up the membrane, and via capacitive distance sensing with our sensors, we can measure the contractile force of the cardiomyocytes here. And indeed, there is more to these impedance-based systems. There is more to be investigated next to contractility of cardiomyocytes, because basically any cell type which adheres to a cell culture substrate and gold electrodes can be measured with this impedance-based technology. So one very advanced and interesting method is so-called impedance spectroscopy. Here, with this method, we can investigate a lot of different effects in cells over time in parallel, because we have a full spectrum of different recording frequencies, and therefore we can detect different things that are going on in the cells. And this method here has been introduced in a high throughput format with the Atlas system. And this system, the Atlas, is being used today for investigating, for example, cancer cells within immuno-oncology or GPCR-mediated signaling, just to name two here. So I would say Nanion, the company, has also come a long way. So since more than 20 years, we develop automated patch clamp systems like the patch liner and the synchro patch, and also the Flexite system, which I could briefly show you. And Kirsty will now explain about her extremely new hot findings. And they used, for example, the CardioExcite system as a diagnostic tool. And therefore, I'm really happy to hand over to Kirsty now, over to Australia. And thank you so much for listening.
Thank you, Sonja. I'm now going to pass control over to Kirsty for her portion of the presentation.
Fantastic. Thank you, everyone. It's a real pleasure to be here and to talk to you about this quite interesting project that we started some time ago. I'm going to preface this with I'm a virologist. I specialize in infectious disease. So this has been a really good learning experience for me about cardiovascular disease, and in particular about the technologies that are available in this field. So at the start, very early on in the COVID-19 pandemic, a lot of patients started reporting that they weren't actually recovering fully from the acute disease. And this was really a patient-led conversation. And unfortunately, there were many problems with sort of gaslighting by the medical community. But eventually, patients coined this term long COVID. And we then, in retrospect, realized that actually a large number of individuals experience this prolonged symptomatic disease or this long COVID. It's now estimated that probably at least 65 million individuals worldwide are thought to suffer from long COVID, which is now commonly referred to as PASC, or post-acute sequelae of COVID-19. Long COVID is quite difficult to define as a condition because it actually is associated with more than 200 different symptoms across multiple organ systems. So many of you may be familiar with the neurological consequences, things like fatigue, brain fog. Some individuals have gastrointestinal problems, some have pulmonary problems, and of course some have cardiovascular problems. And these are just some. When you start talking to long COVID patients, you really appreciate the breadth of symptoms that these individuals are experiencing. Probably most cases of long COVID are most severe within the first 6 months of infection, and some individuals will resolve after that 6 months. But there are some individuals who continue to experience these symptoms 1, 2, maybe even 3 years post-acute infection. And this can actually be quite debilitating in terms of people's quality of life, impairing work, school, everything that they used to be able to do. One of the major problems we have with PASC or long COVID is that we don't really have a good definition of the illness. So the current WHO definition says that this is the continuation or the development of new symptoms 3 months after an initial SARS-CoV-2 infection that doesn't really have any other explanation. So this is incredibly vague. And one of the real priorities for long COVID patients is to have some sort of diagnostic test that's much more objective and can be used to not only validate their experience, but also start to guide treatment in this area. Because unfortunately at present, there's really no treatment available except for symptomatic treatment. So this is a really, really big problem. One of the things that we are starting to appreciate about long COVID is that we talk about this as one condition, but probably in reality, it's actually a bit of an umbrella term, and there's probably multiple sub-conditions within this. So you can imagine that an individual who is experiencing, say, neurological complications of long COVID may have different mechanisms, risk factors, biomarkers compared to somebody who's experiencing, say, pulmonary dysfunction. And this was actually shown really nicely by a study here on the left from Imperial College, where they did widespread proteomics on individuals with different subsets of long COVID, so grouping them by the symptoms. So individuals with fatigue, gastrointestinal symptoms, depression and anxiety, cognitive impairment, and cardiorespiratory symptoms. And what they can see there is that there's actually quite different biomarkers for each of these conditions. So this tells us that probably moving forward, we should stop thinking about long COVID as one condition and start to get a little bit more granular and start to look at different subtypes of the condition. One subtype that we became particularly interested in was individuals with the cardiovascular complications of long COVID. So these are individuals who experience things like chest pain, heart palpitations, but also some individuals who have objective measures of cardiovascular dysfunction in terms of on echocardiograms, ECGs, and so on. This is just a diagram summarizing some of the literature in individuals who are either on the top hospitalized with acute COVID or not hospitalized with acute COVID. And in fact, the literature since we made this slide has absolutely exploded. But what you can see is that, say, individuals who are hospitalized with long COVID, 6 months later, about 5% will experience chest pain, while about 9% will experience palpitations. These cardiovascular symptoms are probably less pronounced in individuals who had a more mild acute COVID-19, but still we see things like excess cardiovascular disease burden up to 5 months post-infection. So again, moving towards a more granular and nuanced understanding of long COVID, the American College of Cardiology actually defined 2 subsets of cardiovascular long COVID. The first they have defined as PASC-CVS, and this is a heterogeneous disorder that may include a range of self-reported cardiovascular symptoms. So chest pain, heart palpitations, post-exertional malaise, fatigue, but not something that is necessarily going to come up on objective clinical diagnostic measures. The other subtype of cardiovascular long COVID is what they've coined PASC-CVD, and this one includes more objective signs of cardiovascular disease, such as myocarditis, ischemia, and arrhythmia. Now, obviously, PASC-CVS is much harder to diagnose because this is really dependent on self-reporting and patient-reported symptoms. So this is something that we wanted to focus on. As Sonja alluded to, there's been really a lot of discussion about what drives long COVID. And there's lots of different theories in this area, but probably one of the best supported is this idea that individuals with long COVID are experiencing some level of chronic inflammation. So we know that individuals with long COVID have increased complement activation, and this can actually lead to thromboinflammation and potentially facilitate clot formation, which can then drive some of the symptoms. We know individuals with long COVID have ongoing neutrophil activity, B-cell memory alterations, increased autoreactivity, and we also know that at least 8 to 10 months post-infection, individuals with long COVID have elevations in pro-inflammatory cytokine levels. It was also recently shown that if you actually take the white blood cells of individuals with long COVID and you look at what they're producing this chronic low level of interferon gamma up to 30 months post-acute infection. Now, we have to ask ourselves, if this chronic inflammation is a feature of long COVID, what is driving it? And there's lots and lots of different theories. Some people believe that this is being driven by viral persistence, so that there is some level of active SARS-CoV-2 replication that's occurring in the body in some privileged site that we can't detect that is driving this disease. The evidence for this is mixed. Certainly, we know that this virus stays around longer than we thought it did, but there hasn't really been concrete evidence of sort of active viral replication in this prolonged phase. The other option is that it's not necessarily the infectious virus that's sticking around, but rather components of the virus that activate the immune response as pathogen-associated molecular patterns. So these are things like persistence of viral RNA, persistence of viral protein, and this is probably much more supported than the persistence of infectious virus. And then, of course, there is the third theory, and this is that long COVID doesn't actually necessarily relate to the SARS-CoV-2 virus in and of itself, but rather that long COVID is characterized by the reactivation of latent viruses that many of us are infected with. So viruses like Epstein-Barr virus, EBV, that causes glandular fever or mono. And what happens in long COVID is that these viruses reactivate into their active state, and this causes chronic inflammation. And we don't really know But what we do think is that in terms of these cardiovascular complications of long COVID, chronic inflammation is really, really important, because we know that a pro-inflammatory response can actually result in the destabilization of atherosclerotic plaques, leading to embolisms, ischemia, and tissue damage. So what we wanted to do is really investigate individuals who had these cardiovascular complications of long COVID and see if we could understand what was causing that, and in particular, understand the role of chronic inflammation in this process. So what we did is a very initial, just proof of principle study, is we collected whole blood samples from 4 individuals who had had SARS-CoV-2 but had had no prolonged individuals, and 5 individuals who had what we call PASC-CVS, so individuals who were self-reporting chest pain or heart palpitations. These were collected approximately 500 days post-acute infection. So this is, quite a prolonged period after their acute infection. These were age and sex matched individuals, and the severity of the acute infection is listed here, although there's very small numbers. So it's really hard to make any conclusions there. What you could see in figure A here is a PCA plot just showing how the gene expression of these whole blood samples grew. And you can see really nicely that the recovered samples, so the samples from individuals who never had prolonged symptoms from SARS-CoV-2, group nicely together, and then the individuals who have long COVID and cardiovascular long COVID are grouping nicely together. And when you start to look at what genes are actually differentially expressed between these two groups, you can see really clearly that it's genes of the immune response. So it's associated with things like neutrophil activation, the humoral immune response, and all this sort of inflammatory gene pathways are listed here. So that was obviously a really promising sign that you could see some level of chronic inflammation in individuals with the cardiovascular complications of long COVID. But of course, these are really small preliminary studies. So we wanted to do this in a larger patient cohort. So what we did is we recruited individuals across 3 sites in Australia, from South Australia, the University of Queensland, and the Mater Hospital in Queensland as well. And we recruited 41 individuals who had recovered from SARS-CoV-2 without prolonged symptoms, and 23 individuals who self-reported ongoing cardiovascular symptoms. The symptoms of our long COVID cohort are just shown here pictorially on the right. So you can see that all individuals had either chest pain or heart palpitations. The majority had fatigue and shortness of breath, which you can sort of imagine goes hand in hand with cardiovascular complications. And then some individuals also had things like brain fog and a loss of smell and taste. Again, these individuals were quite some time after the acute infection, so approximately 400 to 500 days following the acute infection. We did see that in our long COVID cohort, there was significantly more females, and that's consistent with the literature because we know that women, for whatever reason, have a higher risk of long COVID than men. The age was also significantly different between the two groups, with long COVID individuals typically being younger. Again, this is consistent with the literature. And we also saw that individuals with long COVID were more likely to have a moderate or severe acute illness compared to individuals who had recovered disease, where they were more likely to have a mild acute illness. And again, this has been reported in the literature where we know that severity of the acute illness is a risk factor for long COVID. We also had significant site-to-site differences. So just as a note, it means that in all the subsequent analyses, we have controlled for these sex differences, these age differences, and these differences in recruitment site to make sure that we're confident in the validity of our findings. So the first thing we did was very, very simple. We looked at levels of a wide variety of different cytokines in the blood of these individuals using sort of standard multiplex bead-based arrays, so conventional technology. And what you can see here is we've got 2 recovered SARS-CoV-2 groups that are shown in yellow and orange, and then the long COVID groups are shown in red. And if you can see on the graph here, we also have little dashes. These indicate historical samples obtained from individuals prior to the pandemic. So these are levels in individuals who never had SARS-CoV-2, although we do interpret it with a grain of caution because these are historical samples. So really the takeaway message here is there's not much of a difference between the two groups in terms of these standard cytokine measurements. There's maybe a little bit of a difference in interferon lambda and again in interferon alpha, but it's nothing really that striking. And it's also really important to note that a lot of these cytokines were actually at the detection limit of this assay. And one of the reasons we suspect this is happening is because unlike other studies that have looked just 3 months post-infection, we're really looking at sort of over a year post-infection. So we think that that extended period past the post-acute phase is meaning that we're not really seeing many cytokines. So we wanted to explore this a little bit further and see what if we could improve the technology that we were using to detect these cytokines. Would we start to see differences? So this is where my colleagues from the Australian Institute of Bioengineering and Nanion Technology came in, shown here, Matt Trow and Elaine Worthridge. And what they developed was actually a chip that could actually detect cytokine levels down to the femtogram level. So not nanograms, femtograms. And this was all done sort of using nanotechnology. You can design what cytokines you want to put on the chip. And you can see really nicely here, it's all measured with Raman microscopy. And you can see here as if you put cytokines on the chip, they come up as different colors and you can measure that. Whereas if you put other things on the chip, you don't really get a signal. So this chip was initially developed to understand immune toxicity in immunotherapy, but we thought that this was something that we could repurpose for long COVID. So we asked Matt and Elaine to design the chip, and we focused in on just 4 cytokines. Ideally, you could do a larger number of cytokines, but we were sort of limited by the chip technology in terms of how high throughput this could be. So we picked 4 cytokines that in the literature had previously been implicated in long COVID. And what we saw with the chip technology was really quite striking. What you can see consistently is that when we're looking at IL-12, IL-1 beta, MCP-1 or IL-6, they are all significantly elevated in our long COVID cohorts compared to our recovered individuals. And again, if you look for our healthy non-SARS-CoV-2 infected controls, they're way down here. So it's really indicating that long COVID is increasing the trace levels of these cytokines. We actually looked at the correlation levels between these cytokines and also between the time since infection. And in general, we didn't see much of a correlation between cytokine levels and the time since infection, with the exception of MCP-1, which seemed to go down over time. But we did see that these cytokine levels were highly correlated with each other. So if you had high levels of MCP-1, you probably also had high levels of IL-6. So this speaks to, again, a sort of chronic inflammatory phenotype. Now, as I said to you, these are trace level cytokines. So these are very, very low level cytokines. And whilst it's all well and good to see that they're elevated in long COVID patients, we have to then ask, do these have any physiological implications? Could cytokine levels this low actually affect cardiovascular dysfunction and cause the symptoms that these individuals are experiencing? So this brings us to our collaboration with Nanion and our use of the CardioExcite machine. So Sonja showed you the FlexSite plate, which can measure contractility. Here I want to introduce the CardioExcite system, which can measure both contractility and electrophysiology. So the system is also an impedance-based platform, and the cardiomyocytes are adhered on gold electrodes of unique 96-well measurement plates, which reveal extracellular field potentials and also typical beat shapes. So here you can see examples of traces as recorded in commercially available stem cells derived from cardiomyocytes. So what we wanted to do is take cardiomyocytes, stem cell-derived cardiomyocytes that we had grown in vitro, and expose them to these trace-level cytokines and see if that affected their impedance, their contractility, and really cardiovascular function. So this is what we did. And what we did here is we had 3 groups. The first group shown in green is our control group, and these are cardiomyocytes incubated with just media only. The second group is what we call our recovered cytokine mimic. So this is a combination of all the 4 cytokines that we measured, but they are at levels detected in our recovered SARS-CoV-2 patients. And then in red, we have what we call our long COVID cytokine mimic. And these are cytokines, again, these 4 cytokines, but these are at slightly higher concentrations to reflect those elevated concentrations we saw in long COVID patients. And what you can really see here is that these long COVID cytokine mimics result in decreased cardiomyocyte amplitude after 24 hours, upstroke velocity, and increased relaxation velocity. And you see the same thing after 48 hours. So this was really, really clear evidence that these trace-level cytokines, even though they're at a really, really low concentration, can have some sort of physiological function on cardiomyocytes. Now, logically, you would expect that if these trace-level cytokines have an effect on cardiomyocyte function, we should be able to take the plasma of long COVID individuals, place them on the cardiomyocytes, and see a similar phenotype. So this is exactly what we did. Again, we've got our different cohorts here. We have 2 recovered cohorts and 3 long COVID cohorts, and we incubated the cardiomyocytes with the plasma of these individuals for either 24 or 48 hours. And here again, you can see a difference in relaxation velocity, also upstroke velocity, and also amplitude. To see if this was really being driven by these cytokines, we focused in on this difference in upstroke velocity, and we performed these experiments again, but this time in the presence or absence of dexamethasone. So for those of you who aren't familiar with it, dexamethasone is a sort of generic anti-inflammatory that can reduce the inflammatory response, and it's actually used therapeutically in acute COVID for individuals who are presenting in the later stage of disease and have that cytokine storm. So again, you can see exactly what we showed you before. In the absence of dexamethasone, you have this nice decrease in upstroke velocity in the long COVID sera. But if you add dexamethasone, you can actually inhibit that effect. So that is very suggestive that it is something inflammatory in the plasma of these individuals that is driving this phenotype. Now, if you remember what I showed you with those cytokine mimics, the cytokine mimics and the long COVID plasma both affect cardiomyocyte function using this cardioexcite system. But we wanted to be even more sure that we were actually getting changes in cardiomyocyte function. So what we did is after we incubated these cardiomyocytes with the plasma of these individuals, we actually performed RNA-seq and looked at the transcriptomic response. In figure A here, you can see the PCA plot, and you can see that in red, we have the long COVID individuals, and in yellow, we have the recovered individuals. And really nicely, these 2 groups are grouping together, suggesting that they have gene expression patterns in common. And in fact, when you look at the gene expression patterns that they have in common, what you can see is that in our long COVID group, we get upregulation of genes associated with things like angiogenesis, the inflammatory response, coagulation, and hypoxia. So this really suggests that there is something fundamentally different about the long COVID plasma that is driving inflammation and cardiomyocyte dysfunction. Now, if you remember back to what I showed you with the cytokine mimics, we actually had a bit of a cleaner phenotype with the cytokine mimics than we did the plasma. And to me, this probably isn't too surprising because we know that plasma has a lot of things in it aside from just cytokines. So what we wanted to do is see if there was other markers in this plasma that were indicative of long COVID. So to do this, we took our long COVID plasma and we took our recovered plasma and we performed proteomics. What we saw was a whole list of proteins that were upregulated in our long COVID plasma compared to our recovered plasma, and also compared to our healthy controls or individuals who'd never had SARS-CoV-2. The list of proteins is shown here on the left, but this is just some of the interesting proteins shown here graphically. So what you can see is that in the long COVID plasma, we had elevated levels of prothrombin as well as serum amyloid A4 protein. These are actually both implicated in coagulation and blood clots. And this has been one of the defining features of long COVID that was noticed very early on, to the point where there are some individuals who are undergoing therapies to try and basically a plasmapheresis to try and remove clots from the blood to try and reduce their long COVID symptoms. Similarly, we also saw a large number of complement cascade proteins being upregulated in our long COVID group. Once again, this is really consistent with what other people have shown. Insofar as I mentioned to you in the introduction, we know that long COVID is associated with this long-term activation of the complement response. So just to sum up what I've shown you today, really by exploring these cardiovascular complications of long COVID, what we think we've developed or identified is multiple new diagnostic opportunities for long COVID. And as I said to you earlier, this is really, really key because we've got a community of patients out there who are experiencing symptoms, cannot get definitive diagnoses, are sometimes dismissed by the medical community for people who do not believe in long COVID. And so what we propose is that we really have three options here for diagnostics. The first is the nanotechnology, and looking for those trace level cytokines that we can see so clearly increased in our long COVID cohort. The second is using the cardioexcite system and actually looking at cardiomyocyte function. And then the third is the proteomics. Our brief experiments with dexamethasone suggest that maybe for individuals with long COVID, in particular the cardiovascular complications of long COVID, anti-inflammatories are a well-suited therapeutic. And this is something that is currently in clinical trials. There was some research, and there still is ongoing research, that looked at antivirals in long COVID individuals, and this didn't seem to be so promising. So we're all sort of hoping that anti-inflammatories will provide some sort of symptomatic relief. And then finally, I think these data really emphasize that, that PASC is really, it's an umbrella term, and the more we learn about it, the more we appreciate that this has got a lot of different subsets of disease within it, and that we need to consider this in both our diagnosis and our treatment approaches. Finally, I'd just like to thank everybody who made this work possible. This was an incredible collaboration between lots and lots of different groups. I would like to thank all the participants who were really, really supportive of the research, gave feedback on their symptoms, what they were experiencing. Of course, our funders, and of course, Nanion for helping us through this work, and you all for listening. Thank you very much.
Thank you, Kirsty and Sonja, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the lower left corner of your screen, and we'll answer as many questions as we have time for. All right. Sonja, did you
I have one question coming in here.
Perfect.
Does this kind of diagnosis with plasma and functional measurements of cardiomyocytes, has this been done before? Are you aware that?
So no, no one has done this before. And I think that really reflects the fact that as I said to you, I come from an infectious disease background. So for us doing things with cardiomyocytes, with the cardioexcite system, these are things I wouldn't even think about. So certainly people have tried doing experiments where they've transferred plasma from infected individuals onto things like endothelial cells, and they've done some qPCR and seen some inflammation, but nobody's looked at this in the context of cardiomyocytes. And so what this has done for us is open up a lot of opportunities where we now want to try this, not just in long COVID, but in other infectious diseases. What if we could use this to understand the cardiovascular complications of flu, for example, or even more specifically, understand why those cardiovascular complications of respiratory diseases are so much more prevalent in individuals with diabetes. So it really opens up a whole new area of research that we'd never considered before in infectious disease.
And maybe it's also interesting to look at the electrophysiology of the cells, right? You look at the field potential, maybe see something different here in between the ion channel expression or, functionality of the different ion channels. So that's also very interesting, I would say.
Absolutely. There's so much we can do with this system. And I think, when we started this work, we sort of did it as a bit of let's see if it works. And, it's probably not going to work and we're not going to see anything interesting, but let's do it anyway. And so we were really surprised at how obvious and clean the results were. I think, as I said, there's a lot more to be done in this field. And I think it really speaks to the benefits of interdisciplinary work. As I said to you, I'm a virologist, and so I started this as a virology perspective. But in doing so, and to answer these questions, we've had to bring cardiologists on board, nanotechnologists on board, clinicians, mathematicians. It's been really that interdisciplinary team that's let us get these really great insights.
Mm-hmm.
Great. We have another question here that popped up. This one asks, is it surprising that you see inflammatory cytokines increased, including IL-6, but you don't see elevated C-reactive protein in your proteomic analysis?
Yeah, this is something that we actually discussed a little bit, and it might reflect the limitations of the proteomic analysis. So unlike the nanotechnology that we use, the proteomic analysis has a much higher detection limit. And in fact, it's when you go to a clinician and you get your CRP levels measured, that's much more sensitive than what we did with the proteomics. So we suspect that probably there is CRP elevated in these individuals. It's just with the proteomic technology, it's not sensitive enough to detect it.
Great. Thank you. Right. Our next question that we have asks, is there any indication on the ion channels involved in the alterations of the electrophysiological profiles observed following the administration of the cytokine cocktail?
Yeah, great question. This is something we really need to do. We haven't looked at this. Even, people have done this sort of work with higher concentrations of cytokines and they have seen changes, but whether or not we will see them with those trace level cytokines, I don't know. But yeah, it's definitely something that we flagged to follow up because I think it's really key.
Excellent. Thank you. Right. If anyone else has any questions for our speakers here, feel free to submit them. While we're waiting, Sonja, Kirsty, do either of you have any comments for our audience today?
I think from my perspective, it's just really appreciating how prevalent long COVID is and how we still have such a poor understanding of it. This was something I spent a lot of time working in pandemic preparedness. And so prior to COVID-19, we had these great some pandemic preparedness plans, mainly focused on outbreaks of influenza. And nowhere in any of the pandemic preparedness plans did we consider this post-viral illness and what effect that would have on society, what effect that would have on healthcare utilization. And so I think that COVID-19 has taught us a very valuable lesson in this regard. And we are starting to really appreciate the long-term effects that viral infections can have. Whether or not this will apply to the next pandemic, I don't know, but I think it's really worth investigating, because if it does, and it's not just a SARS-CoV-2 specific phenomenon, which it doesn't seem to be, we need to put this into our pandemic preparedness plans.
Yeah, I agree. And I mean, the story you presented is so interesting and so new. I was really happy to work together with you, at least on some part of that. So yeah, let's see how that proceeds. And yeah, I can just thank you for staying up late in Australia.
It's all right. It's early morning now. It's passed over into the next day. So, no, and I think that, the next step that we're doing, again, with the support of Nanion, is really to go into the diagnostics of this and look at the potential of creating a diagnostic and prognostic test for individuals with long COVID. So watch this space.
Great. Well, thank you both for your time today and your important research. We would also like to thank LabRoots and our sponsor, Nanion Technologies, for underwriting today's educational webcast. Before we go, I'd like to thank our audience for joining us today and for their interesting questions. Questions that we did not have time for or those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thank you. Bye-bye.